In this video, we will study about DNA replication and the semi-conservative mode of replication of DNA and the replication in prokaryotes. So first of all, what is DNA replication? DNA replication is the process by which DNA will copy itself inside a cell. That process is called as DNA replication. It means that one DNA molecule will form two DNAs. So that is the process of the DNA replication. Now, Watson and Crick, they gave the double helix model of DNA in the year 1953, in which they described the structure of DNA, that it is a double-stranded molecule and it is composed of sugar, phosphate and nitrogenous bases. The two strands are connected to each other with the help of hydrogen bonding. After that, Meselson and Stahl experiment in 1958 proved that the mode of replication of DNA is semi-conservative in nature. Now, there can be three modes of replication that are possible. One is conservative mode of replication, another one is semi-conservative, and dispersive mode of replication is the last one. So, in conservative mode of replication, suppose there is a DNA strand. This is parental DNA strand in red and the blue one is the new DNA strand. So in conservative mode of replication, what happens is the newly synthesized DNA, it is completely new, whereas the parental strand, it is conserved totally. It means that the parental strand remain as it is and the newly synthesized DNA, it is completely new. Whereas in semi-conservative mode of replication, what happens is, suppose these are the two strands of DNA. They get separated into two strands and each strand will act as a template and will synthesize its own strand. So in this way it is semi-conservative. Yani ki the newly synthesized DNA, it has one parental DNA and one will be the newly synthesized DNA. So this is semi-conservative model. In dispersive mode of replication, what happens is some part of the DNA is conserved, whereas the other part, it is totally new. Okay. So, Meselson and Stahl proved that breaking of hydrogen bonds between the two strands will give rise to two separate DNA strands, and these will act as a template for the synthesis of new DNA strands. So, this is the semi-conservative model. How did they prove the semi-conservative model? So they did an experiment on E. coli. So as we all know that DNA has nitrogenous bases and that nitrogenous bases has nitrogen in it. So nitrogen basically has two isotopes. One is the N14 isotope, another is N15 isotope. N15 is heavy and N14 is light. So the normal DNA, it has N14 type of nitrogen, but Meselson and Stahl, they took E. coli and grew it in N15 media for several generations, due to which the DNA in the E. coli gets labeled with the heavy isotope. It means N15. The DNA becomes N15. So this red is the N15 DNA. Then they took this E. coli and grew it in N14 medium for one generation, due to which what happens is the N15 DNA, it remains as it is, the parental DNA, and the newly synthesized DNA, it has N14 in it, the light nitrogen, it has N14 in it, and two hybrids are formed. Then they grew E. coli for another generation in N14 media and saw that there were two hybrid DNA and two will be the light DNA. Two light DNA are there and two hybrid DNA are there. Why does it happen? Because parental DNA, it gets separated, this N15 gets separated, and the newly formed DNA, it has N14 in it, so two hybrids are formed. Similarly, in the next generation, what happens is all these strands, they separate out from each other, and newly synthesized strands, they will have N14 in it, so two hybrids will be formed and two light DNA will be formed. So they checked the hybrids and the light and the heavy DNA using cesium chloride centrifugation. in which the heavy DNA was found at the bottom, forming a band at the bottom of the cesium chloride, whereas hybrid form of DNA, it will form a band in the middle of the cesium chloride, whereas the light DNA which is formed, it will form a band at the top of the cesium chloride density gradient gel. So they found that the after several generations of N15 media, they found that the DNA gets labeled with N15 and the band is formed at the bottom of the cesium chloride. And after one generation of growing in N14 medium, the band was observed in the middle. It shows that the nitrogen is N15 and N14. It means that hybrid is formed after one generation. And after that, after one more generation, they observed that the bands are formed. One is formed in the middle and one is formed at the top. It means that half of the DNA formed were hybrid and half of the DNA formed were light. So using this experiment, they proved that the mode of the replication, it is semi-conservative in nature. Since half of the parental DNA, it was conserved after each generation, so it was semi-conservative mode of replication. Now, origin of replication. What is origin of replication? It is a point from where the replication will initiate in the DNA. So the origin of replication is, it is an AT rich region. Yani ki the A and T bases, they are found in this region. So what is a replicon? A replicon is the unit of DNA replication. A unit of DNA replication, it is the region of DNA which is replicated by a single ORI. यानी कि एक सिंगल ORI से जितना भी पोर्शन ऑफ DNA रेप्लिकेट होगा वो पूरा का पूरा क्या होगा, एक रेप्लिकॉन होगा. सो अ रेप्लिकॉन इज अ यूनिट ऑफ DNA replication, region of DNA which is replicated by a single ORI. Okay. The DNA replication is bidirectional, yani ki it succeeds in two direction because the polarity of DNA strands are opposite to each other. One is in 5' to 3' direction, another is in 3' to 5' direction. So the DNA replication is also bidirectional, except in the case of F plasmid. In F plasmid, the DNA replication is unidirectional. So what is the difference of origin of replication in eukaryotes and prokaryotes? Because eukaryotes have vast DNA, so there are multiple origin of replication in eukaryotes, and the origin of replication is called ARS, yani ki autonomously replicating sequence. Whereas in prokaryotes, there is a single origin of replication which is called as ORI region. In eukaryotes, it is multi-repliconic. Multi-repliconic means that there are many replicons. There are many replicons. Yani ki there are many DNA which are synthesized from many origin of replications. So it is multi-repliconic. Whereas prokaryotes, they are monorepliconic. Yani ki the whole complete DNA is synthesized in a single replicon with the help of a single ORI. So it is monorepliconic. In eukaryotes, the replicons are smaller in size, whereas in prokaryotes, the replicons are larger in size because there is a single replicon. So replicons are larger in the case of prokaryotes. Now, autonomously replicating unit, ARS, in the eukaryotes, it is composed of 200 base pair 80 rich region, whereas ORI, it is composed of 245 base pair long 80 rich region. Now, there are certain conserved boxes which are present in the origin of replication. What are these conserved boxes? These conserved boxes will help in the initiation of the replication. In the case of prokaryotes, in ORI, there are two types of conserved boxes that are seen, that are 13 mers and 9 mers. Whereas in eukaryotes, there are four different conserved boxes which are present: A, B1, B2 and B3. So these four conserved boxes are present in the case of eukaryotes and two conserved boxes are present in the case of prokaryotes, 13 mers and 9 mers. 13 mers means they are 13 base pair long and 9 mers means they are 9 base pair long. This is ORI site in prokaryotes. These are 13 mers and these are 9 mers which are present. And this is 245 base pair long. Now there is one thing, 9-mers is the binding site for DNA A protein in the replication process, and 13-mers, what is 13-mers? Here the melting of ORI occurs. Melting of ORI means that the DNA will form a replication bubble from here. So here melting starts and here DNA A protein will bind. So this is the significance of 9-mers and 13-mers in the DNA replication process. Now what is a replication bubble? So as we all know that DNA replication will begin by breaking of the hydrogen bonds between the strands at the ORI, यानी कि at the origin of replication the hydrogen bonds will break between the two strands of the DNA, and this breaking of hydrogen bonds means melting. It means the DNA is melting. So this melting will give rise to the structure called as replication bubble, to a structure called as replication bubble. For example, this is a DNA. So this will melt at the origin of the replication, and this is replication bubble. Here breaking of the hydrogen bonds will occur at this site. So this is called as a replication bubble. This is a replication bubble. So it is the origin of replication, and here the melting will occur, the breaking of the hydrogen bonds will occur, and it will separate out. So in this, as we can see that there are two fork kind of structures which are present, and these are called as replication forks. So in one replication bubble, two replication forks are present, one this one and one is this one. So the replication will proceed in two directions, one in this direction and another one will be in this direction. So the DNA replication is bidirectional. Also there is one leading strand and one is the lagging strand. What does this mean? Since we all know that the two strands of the DNA has different polarity, one is in 5' to 3' direction, another is in 3' to 5' direction, and the DNA is synthesized only in the 5' to 3' direction. So this is the replication fork, and from here replication will begin. The strand, the template strand which has the polarity 3' to 5', it will be synthesized continuously in the 5' to 3' direction. But the DNA strand that has the polarity 5' to 3', it cannot be synthesized continuously, because DNA strands can only be synthesized in the 5' to 3' direction. So it will be synthesized in the opposite direction, that of the replication fork, due to which the DNA synthesis, it is discontinuous. For example, the RNA primers, they get attached here at the sites, and the replication will begin in the opposite direction here. So the replication in the lagging strand, it is discontinuous. It means that it occurs in fragments. It occurs in fragments, whereas in the case of the leading strand, the replication is continuous. So this is the concept of leading and lagging strand. Now what is the difference of leading and lagging strand? The leading strand, it polymerizes in the direction of the movement of the replication fork, whereas the lagging strand is polymerized in the opposite direction to the movement of the replication fork. Here we can see that the leading strand, it is synthesized in the direction of the replication fork. The replication is the replication bubble, it is moving in this direction. So the DNA is synthesized in the direction of the replication fork, whereas the DNA synthesis in the lagging strand is in opposite direction to that of the movement of the replication fork. So one difference is this. Another difference is the leading strand is polymerized continuously, whereas lagging strand, it is polymerized discontinuously. यानी कि ये fragments में form होता है और ये continuously in one go it is formed, and here Okazaki fragments are formed. What are Okazaki fragments? Okazaki fragments are basically the fragments which are formed that I showed you. These fragments which are formed, these are Okazaki fragments, whereas in this case there are no fragments. So this is the difference between the leading and the lagging strand. Now, what is prokaryotic DNA replication? So, prokaryotic DNA replication, the whole process, it can be divided into three main steps, that are initiation, elongation, and the last one is the termination. So, first I'm telling the different factors which are involved in these steps. In initiation, we require DNA A, DNA B, DNA C, DNA gyrase, and SSB proteins. In elongation, we require DNA polymerase 1, DNA polymerase 3, DNA G, which is primase, and SSB proteins, DNA ligase, and DNA gyrase. In termination, we need topoisomerase 4. And there are removal of TUS proteins from the TER sites. The first step in the initiation of replication is the formation of replication bubble. Now, how the formation of replication bubble occurs? There's a protein called as DNA A protein. This DNA A protein, it will get activated in the presence of ATP and will bind to the 9-mer region, and its binding will cause the melting at the 13-mer region. For example, this is the 9-mer region, and here the DNA A protein will bind and it will cause the melting at the 13-mer site, and due to melting there will be the breaking of hydrogen bonds and two DNA strands will be separated. So this is the first step, that is the formation of the replication bubble by DNA A protein. After the formation of replication bubble, the second step is binding of the SSB proteins. Now, SSB proteins are single strand binding proteins. These are single strand binding proteins. What is the function of SSB proteins is that they will bind to the separated DNA strands and will restrict them from re-annealing. These SSB proteins will restrict the separated DNA strands from re-annealing to each other. So these SSB proteins bind to the separated strands. After that, the third step is the binding of the DNA B and DNA C. DNA B and DNA C. So DNA A protein, which is already present on the template strand, it will recruit DNA B. DNA B is the helicase. DNA B is helicase and it will cause the unwinding of the DNA. So DNA B is helicase and DNA C is helicase loader. DNA C, यानी कि the helicase loader, it will load the DNA B on the strand. So DNA A recruits DNA B at both the strands, which is loaded on both the strands with the help of DNA C. This is step number 3. And after the loading, DNA C, it will dissociate from the DNA B after loading is completed. DNA B, it is a homohexameric ring-like structure. It is a homohexameric ring-like structure just like this. And DNA C, it is a helicase loader, will work using ATPase activity. After this, the DNA B helicase, it will further melt, cause the melting of the DNA and the bidirectional movement of the replication bubble. So DNA B helicase will conduct the melting. After the this step, in step 5, DNA G primase. DNA G primase protein, it is involved. What is DNA G primase? It is basically a type of RNA polymerase. DNA G primase, the function of DNA G primase is that it forms primer. It forms RNA primer on the DNA strands. It forms RNA primer at both the strands, and after that it will detach from the DNA. Second part of the DNA replication is elongation. In this, DNA polymerase enzyme is involved. So the step 1 of elongation process is that DNA polymerase will recruit DNTPs, which are dideoxynucleotide triphosphate, at the catalytic site. So DNA polymerase, it will recruit the DNTPs and will facilitate the nucleophilic attack from the 3' OH of the pre-existing nucleotide to the alpha phosphate of the new DNTP. Like this is DNTP, this is new DNTP which is there, and alpha phosphate, it is the phosphate which is directly attached to that DNTP, and this is the beta phosphate, this is the gamma phosphate. So what does DNA polymerase do? It will facilitate the nucleophilic attack of the 3' OH that will be on the pre-existing nucleotide to this alpha phosphate of that new DNTP which is present. And like this, it will lead to the bond formation between the newly synthesized new DNTP and the pre-existing DNTP. So step 2 will be, due to nucleophilic attack, there will be a release of pyrophosphate. Pyrophosphate means 2 phosphates will be released from this DNTP. Why? Because the 3' OH of the pre-existing nucleotide, it makes an attack on the alpha phosphate and makes a phosphodiester bond with the alpha phosphate. So these 2 phosphates, these are released as pyrophosphate. These are released as pyrophosphate, and there will be a formation of phosphodiester bond in step 2 between the pre-existing nucleotide and newly recruited DNTP or DNMP. NMP, why? Because deoxynucleotide monophosphate it is, right? Because 2 phosphates are already released. Step 3, there will be a hydrogen bond formation between the template and the daughter strand. So once the newly recruited DNMP, it is incorporated inside the newly synthesized DNA, there will be a formation of hydrogen bond between the template and the daughter strand. So this is the process of elongation, in which first DNA polymerase will recruit DNTP and there will be a formation of phosphodiester bond, and after the formation of phosphodiester bond there will be hydrogen bond formation. Now the DNA polymerase which is involved in the polymerization process in the formation of the DNA strand, the main DNA polymerase, it is DNA polymerase 3. It is DNA polymerase 3. This DNA polymerase 3, it is a holo enzyme, yani ki it has multiple subunits that will work cooperatively. It has multiple subunits that work cooperatively. What are these subunits? There are mainly 3 subunits which are there. Core DNA polymerase, which actually does the polymerization process. Sliding clamp, which is responsible for the sliding of the core DNA polymerase 3. And there is a clamp loader, which is responsible for loading this sliding clamp. So there are 3 different subunits of the DNA polymerase 3 which will work cooperatively. Now there are subunits of these 3 subunits as well. In core DNA polymerase 3, alpha, beta and theta, these 3 subunits are present. In sliding clamp, there is only one subunit, that is beta subunit. Whereas in clamp loader, there are 5 different subunits which are present. Gamma, chi, psi, delta and delta naught. These 5 subunits are present in clamp loader. What are the functions of these 3 different parts of the polymerase 3? The core DNA polymerase 3, it is basically responsible for the 5' to 3' polymerization activity and 3' to 5' exonuclease activity. It means that it will synthesize the DNA and it is responsible for the proofreading of the DNA. We will discuss about it later. The sliding clamp, it is basically a dimer. It is basically a dimer shaped molecule, and it is a ring shaped dimer like this. And the clamp loader, it is responsible for the loading of the clamp. There is another subunit which is present, that is tau subunit. This tau subunit, it is a dimerizing subunit for this core DNA polymerase 3, it dimerizes with it. And also there are 5 different DNA polymerases which are found in prokaryotes. DNA polymerase 1, DNA polymerase 2, DNA polymerase 3, 4 and 5. And in DNA replication process, mainly DNA polymerase 3 is important, and apart from this, DNA polymerase 1, it also has a part in DNA repairing. Now the next step of the elongation, it is the removal of the RNA primer. Now as the primer which is there, it is RNA primer, it has to be removed because DNA and RNA has different base pairs, so the RNA primer must be removed and replaced by the DNA. So this is done by DNA polymerase 1. The RNA primer is removed by 5' to 3' exonuclease activity of the DNA polymerase 1. Gaps which are formed is removed by 5' to 3' of polymerization of polymerase 1, but cannot seal the last phosphodiester between the nucleotide. It means that the DNA polymerase 1, it will remove the RNA primer and will synthesize the DNA, and thus replacing the RNA which was there, but the last phosphodiester bond which remains, it cannot seal that. So there is a nick formation which occurs at the last phosphodiester bond. How does this nick formation, how does this nick get sealed? It gets sealed by the DNA ligase. So DNA ligase seals the nick. So the next step is the DNA ligase, the nick sealing. It has the ability to seal the nick which is formed by the formation of the phosphodiester bond between the 3' OH of the one nucleotide and 5' phosphate of the other. The last step in the elongation process, it is proofreading. Now as DNA synthesis, it occurs at a very fast rate, यानी कि 1000 nucleotides per second are synthesized in the DNA polymerization. So there is a very high chance that a wrong nucleotide will be incorporated. So the incorporation of wrong nucleotide, it may lead to the mutation, and hence it has to be removed. So this is done by DNA polymerase 3. This is done by DNA polymerase 3. Because DNA polymerase 3 has a property of 3' to 5' exonuclease activity, by which it can remove the nucleotide which is wrongly incorporated. It will remove them and will replace them with the right one with the help of 5' to 3' polymerization activity. So this is done by polymerase 3. Proofreading is done by polymerase 3. Proofreading will occur simultaneously with the polymerization. Okay, so it will occur simultaneously with the polymerization. These both processes, proofreading and polymerization, they will occur simultaneously. The last step of the DNA replication is the termination. Now, how does termination occur? Basically, a termination region is present in the DNA, and this termination region has ter sites. This has ter sites which is 23 base pair long, and there are 7 to 10 ter sites which are present in the DNA. On these ter sites, TUS proteins are present on these. The TUS proteins bind to the ter sites. So these proteins which are present, TUS proteins which are present, they must be removed for the replication to complete, because this halt the process of replication and DNA cannot be replicated completely because of the presence of the TUS proteins. So these proteins have to be removed to complete the process of the replication. And also because of the uncoiling of the DNA during the replication process, because of the unwinding of the DNA, there is a super coil which gets accumulated at the end of the DNA, because the DNA gets unwind and at the end there is a super coil which is present. So this will also lead to the halting of the replication. So these two steps are responsible for the halting of the replication processes, that is the presence of TUS proteins and the supercoiling which is present, and these two factors have to be removed for the replication to complete. Due to supercoiling, a structure which is called as catenin, it is formed. This structure, it is supercoiled. The catenin structure, it is resolved by topoisomerase 4. it will remove the supercoiling. This enzyme, it removes the supercoiling of the DNA. And also the TUS proteins, they are removed by the helicase and the DNA polymerase 3. So the supercoiling gets removed by topoisomerase 4 and TUS proteins are removed by helicase and DNA polymerase 3. So as these two factors get overcome by these enzymes, the replication can continue and the termination region gets fully replicated and the catenin gets resolved, and this is called as decatenation. And finally the fully replicated DNA, it is formed, and after the formation of fully replicated DNA, the separation of the replicated DNA will occur, and this is how replication is completed in prokaryotes.